Ragsmed. Today we will continue the part 2 of mechanisms of cell injury. In the previous video, yesterday I have already covered 3 topics, that is 3 mechanisms of cell injury, that is ATP depletion, mitochondrial damage and loss of calcium homeostasis. So today we will see the 4th and 5th mechanism of cell injury, that is defects in membrane permeability, and 5th one is free radical injury. So first let's see the 4th mechanism, that is defects in membrane permeability. So the consequences of membrane damage are mitochondrial membrane damage, plasma membrane and lysosomal membrane damage. As we have already discussed about the mitochondrial membrane damage, that is, it is the second mechanism of cell injury. So apart from this, we will discuss the plasma membrane as well as lysosomal membrane damage. So let's start our topic. Here in this figure we can see this is a cell with nucleus, mitochondria, cell membrane and lysosome. The cell is bounded by cell membrane. Mitochondria has also double-layered membrane and the lysosome is also bounded by lysosomal membrane. So now any kind of injury can affect any of these membrane. If there is cell or a plasma membrane injury, then there will be formation of gaps in it. So through these gaps, all the cellular contents or plasma will leak out of the cell and ultimately the cell will die. We have already discussed about the mitochondrial damage earlier in the second mechanism of cellular injury. So the last one is lysosomal membrane damage. Since lysosomes are known as suicidal bag, it normally remains intact. But if they rupture, then there will be defect in the lysosomal membrane, then holes will be present in the lysosomal membrane, and through these holes all the contents of lysosome will come out in the cytoplasm and ultimately degrade the cell. As we have already covered the mitochondrial membrane damage in the second mechanism of cell injury, so now we will directly jump into the plasma membrane as well as lysosomal membrane damage. So let's start about the plasma membrane damage. So due to loss of osmotic balance, there will be efflux of fluids and ions, and it will lead to loss of cellular contents and finally metabolites that are vital for reconstitution of ATP. Now let's see about lysosomal membrane damage. So lysosomes contain enzymes such as RNase, DNase, proteases, phosphates, glucosidases and cathepsins. And if there is damage in the lysosomal membrane, then there will be formation of holes, and through that holes there will be leakage of their enzymes into the cytoplasm, due to which there will be enzymatic digestion of proteins, RNA, DNA and glycogen, and ultimately the cell will go through necrosis. So this was all about the 4th mechanism in cell injury, that is defects in membrane permeability. So now let's come to 5th mechanism of cell injury, that is free radical injury. So as we know, free radical injury is also known as ROS, that is reactive oxygen species. So in the mitochondria, oxidative phosphorylation takes place. So what is oxidative phosphorylation? It takes place in the inner membrane of the mitochondria. As we know that O2 enters into the inner membrane of mitochondria and ultimately gets converted into water. During this process, ATP are released at various intervals. So this is not a direct process. There are many intermediates which are formed during this process. So what are those intermediates? Those are superoxide ion, hydrogen peroxide and hydroxyl ion. And ultimately it gets converted into water. Now question arises, since these intermediates are formed in all our cells, then why are they not damaging the cells? Since these are reactive oxygen species, that is free radical. So for this the cell has a backup plan to eliminate them in the form of enzymes such as superoxide dismutase, glutathione peroxidase and catalase. So these enzymes eliminate those free radicals and there is no damage to the cell. Now let us see the last step of conversion of H2O2 to OH, that is hydroxyl ion. So how it is converting? Now if the conversion takes place in the presence of iron, then it is known as Fenton reaction. And if the conversion takes place in the presence of oxygen, then it is known as Haber-Weiss reaction. Now let's see the theory of free radical injury. So normally generation of ATP is done by oxidative process where oxygen, that is O2, combines with hydrogen atom and water is formed. This reaction of O2 to water involves 4 electron donation in 4 steps, involving transfer of 1 electron at each step. So now let's see its generation of free radical injury. So these free radicals are intermediate chemical species having a single unpaired electron in its outer orbit. The unpaired electrons are highly reactive and attack and modify inorganic or organic chemicals such as lipids, proteins, carbohydrates and nucleic acids. So these are generated within mitochondrial inner membrane, and normally generation of ATP is done by oxidative process, that is O2 combines with hydrogen atom giving water. So as we have already discussed about the Fenton and Haber-Weiss reaction, when the conversion of hydrogen peroxide takes place in the presence of iron, then hydroxyl ion is formed. So this is the reaction of Fenton, and when the reaction takes place in presence of oxygen from hydrogen peroxide to hydroxyl ion, then this is known as Haber-Weiss reaction. So the Fenton reaction is involved in free radical generation when iron is converted from ferrous to ferric form. So what is Haber-Weiss reaction? It leads to generation of free radical during normal metabolic process. So coming on the types of free radicals, it is generally of 3 types depending upon the number of electron transferred. So number 1 is superoxide oxygen and it has 1 electron. Number 2, hydrogen peroxide H2O2, it has 2 electrons. And number 3 is hydroxyl radical, that is OH minus, which has 3 electrons. And other free radicals are Nitric oxide, that is NO, it is a chemical mediator formed by various body cells such as endothelial cells, neurons, and macrophages, etc. Second is halide reaction such as chlorine or chloride or bromine or bromide. So, what are the causes? They react with and damage proteins, lipids, carbohydrates, and nucleic acids. Lipid peroxidation in cell membranes, oxidation of amino acids and proteins resulting in fragmentation, and protein-protein cross linkages are the causes of free radical injury. So how these are removed? Three enzymes are present such as superoxide dismutase, glutathione peroxidase, and catalase. Apart from these, there are few enzymes such as vitamin C and E, transferrin, ferritin, and ceruloplasmin. Iron and copper catalyze free radical formation. So the sequence of intermediate is shown here. From oxygen it gets converted into the superoxide ion, that is O2 minus. From superoxide ion it gets converted into hydrogen peroxide, that is H2O2. From hydrogen peroxide it gets converted into hydroxyl ion, and from hydroxyl ion it gets converted finally into water. So superoxide ion, hydroxyl ion, and hydrogen peroxide, these all are the intermediates formed. So now here we can see the Fenton's reaction. So if the conversion takes place in the presence of iron, then this is known as Fenton's reaction. Whereas if the reaction takes place in the presence of oxygen, then it will be known as Haber-Weiss reaction. So the superoxide dismutase converts O2 to hydrogen peroxide. Glutathione peroxidase converts hydrogen peroxide to hydroxyl ion, and catalase converts hydrogen peroxide to water directly, and all these enzymes are formed in the inner surface of mitochondria. Coming in the conclusion of defects in membrane permeability, so what we have discussed is that there are three membranes such as plasma membrane or cell membrane, mitochondrial membrane, and lysosomal membrane. So if the plasma membrane gets disrupted, then there will be formation of holes, and gradually all the cellular or plasma contents will leak out, causing the death of a cell. As we have already discussed about the mitochondrial membrane, so we will skip it. So now coming on the lysosomal membrane, same process occurs here. If the lysosomal membrane gets disrupted, then there will be formation of holes, causing the leakage of enzymes from the lysosomal membrane, as it is known as suicidal bag. So all the enzymes come out in the cytoplasm, causing the necrosis of a cell. So now let's see the summary of free radical injury. So as we have already discussed, there are three intermediates formed during the conversion of O2 to water. So what are those intermediates? They are superoxide ion, that is O2 minus, hydrogen peroxide, that is H2O2, and finally hydroxyl ion, that is OH minus. So these are the intermediates, or also known as free radical, which are formed during the conversion of O2 to water. So this can harm the cell, but the cell has backup plan to eliminate these free radicals with the help of few enzymes such as superoxide dismutase, glutathione peroxidase, and catalase. So all these enzymes help to eliminate these free radicals, and finally there will be no damage to the cell. So this was all about the 5 mechanisms of cell injury. Now let's see some questions. So the number 1 question is, deadliest free radical is what? A. Hydrogen peroxide. B. Superoxide ion. C. Hydroxyl ion. D. peroxynitrite. So drop your answers in the comment section. Number 2. Fenton reaction leads to free radical generation when A. Radiant energy is absorbed by water. B. Hydrogen peroxide is formed by myeloperoxidase. C. Conversion of ferrous iron to ferric iron. D. Nitric oxide gets converted into peroxynitrite. Number 3rd question. Which of the following is not a free radical scavenger? A. Glutathione peroxidase. B. Superoxide dismutase. C. Catalase. D. Xanthine oxidase. So the fourth and last question is that enzymes that protect the brain from free radical injury is A. Myeloperoxidase. B. Superoxide dismutase. C. MAO. D. Hydroxylase. So please comment down in the comment section the answer of all these questions from 1 to 4 to check whether those answers are correct or not. So this was all about the mechanism of cell injury. We have already completed in a simplified way. So drop down your doubts in the comment section, and please don't forget to like, share and subscribe with your friends. Till then, stay tuned, stay connected and stay learning. Thank you.